I'd like to call upon Dr. Jozef Gecz to please join me, along with Dr. Carlos Sala. Both of them are good friends. We've known each other for years. I wasn't diligent, not as much as Professor Guerini, and I didn't prepare an initial slide because, as I said at the very beginning, I don't work on PCDH19. What I would like to say, however, and what was said before by one of the speakers, and mainly Dr. Marini, is that it's important to understand how PCDH19 works. She says you don't have the translation. I hope you do. Well, what we're going to discuss now is a molecular study of genes that are involved in diseases and pathology such as this, because there might be other inputs, other insight for possible therapy once we understand the role played by these genes and proteins within the CNS.
Thank you very much, Patricia. Thank you, Francesca, Manuela, and everybody else who has been involved in bringing people together, either people who are immediately and intimately involved looking after these girls, and also people who are more professionally involved in trying to help to make it as easy as possible. So today I'm going to split the talk in three parts. The beginning is going to give you a little bit of an introduction, maybe a little bit of genetics, and perhaps some of these items or areas we discussed, you may have heard already aspects of it in previous speakers. Then I will tell you a little bit about our work, how we try to understand the molecular mechanism in this disease, so we can perhaps help these girls to cope better and improve their life. And towards the end, I would like just to discuss our findings in the context of what is already known. This work I'm going to show you is not published, so I would rather call it a hypothesis or something we believe in, but it still needs to be rigorously assessed by our peers. And of course, it's still quite a way to go. That's it. So my interest for many years was on the human X chromosome, and it's one of the chromosomes which is for some reason very busy with genes which are important for the normal function of the human brain. It contains only about 5% of the human genome, and as you can see on the left-hand side, that's how it looks. The smaller one next to it, it's the Y chromosome, which is somewhat important, but probably not as much as the X chromosome. So this is a typical X chromosome family, and in these families we see the boys are affected. The girls usually transmit the disorder, but sometimes, as you can see in the middle of the pedigree, the girls can be affected as well. So when we came across, of course, EFMR, as it was published, and as we worked with our colleagues Ingrid Scheffer and Sam Berkovic in Australia, we found several of these families, and here we obviously saw exactly the opposite. So the boys were the ones who transmitted and were apparently okay, and the girls were the ones who had the difficulties. But not always. Sometimes the girls really, like in this pedigree, you can see they are mothers, they look after children. So not all of them were similarly severely affected. And that's, you heard about that today. The spectrum of the severity in these families is amazing, and more patients we're finding, more and more we learn how wide it is. So it is really from the very severe form to almost we call non-penetrant, like these individuals can live without the normal function of this gene, which is in a way amazing by all means. So it was quite amazing that a year after, and this is just a spectrum of the families, as you can see they are huge, and we originally believed that this is likely going to be the case, that we're going to see of these families, but really what the French group found, and they found it through studying a boy with epilepsy, What they found, he was very unusual. In a way, he had a problem with one chromosome in a part of his cells, which made him look like a girl in a way. So he was a mosaic. So some of his cells have a normal genetic composition of the X chromosome, and the other cells had the region of the protocatenin 19 missing. And that created a situation like we see in normal girls, that 50 and 50% on average have the right or the chromosome with the mutation. And based on that story, really everything else unfolded. And we have these huge numbers of patients, which are usually isolated cases. And again, girls primarily affected. To my knowledge, this is the only boy, but correct me if there is another one. This is really the only boy currently known in literature with mutations in this gene. What is also important, and what Carla already mentioned, is that in many cases, in isolated cases, the mutations happen de novo, so you don't see it in a parent. So father doesn't have it, mom doesn't have it, and suddenly there is a girl which has it. She can have it in all her cells, or she can have it in part of her cells. And of course, and then it's crucial for the understanding of her clinical presentations. And then we have situations where the mutation might be inherited from an unaffected father in isolated cases. So we see more and more of these on the right-hand side, this type of girls, rather than those big families which we saw originally in our studies. So this is X chromosome. There are more than 110 genes known to cause different forms of intellectual disability. And probably in about 50% of cases, there is also epilepsy. I'm just showing it because to tell you that what we see in epilepsy due to the protocatenin 19 gene mutations is not in a way unique. We see for many of these genes very broad clinical presentations. These genes have different functions. And the fact that you have one mutations and different outcomes is illustrated by that little picture on the bottom right-hand corner, when really you see different phases or different clinical outcomes as a consequence, often of identical genetic mutation. So along those lines, that's not really new. And also, it's not new that the spectrum of the function of these genes is really diverse. These are different molecules. That's like in a society, if we compare genes to our society. Some of them are truck drivers, some of them are computer scientists, some of them are regulators, some of them are just sweeping streets. So they do more or less complex functions, and based on that we see different outcomes. So we looked into this to some extent, and when we compared all genes involved in intellectual disability or epilepsy, and probably you can put about 800 together. So we're talking today about one gene, but there are probably more than 800 currently known, and we estimate that we are going to see twice as many. So on average, maybe, I mean, it's still speculation, we may end up with 2,000 different genes involved in learning disabilities and epilepsies. And you can see that there is a sizable overlap where these genes or mutations in these genes can cause different types of outcomes in terms of epilepsy and intellectual disability. In terms of function of these molecules, there is the interesting difference. In learning disabilities, the genes involved are mostly the regulators. These are the transcription factors. So these are the molecules which sit higher in the hierarchy and tell many different other genes what to do, how to do, and when to do. While the genes which are involved in epilepsy are more the effectors. So those are those molecules like ion channels or others which have more dedicated to a specific function rather than have a huge regulatory function. But that's perhaps a slight simplification of the complexity. But it's interesting. But also, as you can see, the repertoire of different molecules involved in these different disorders is quite amazing. And I'm saying it also because we need to understand this when we try to fix this problem. All these genes somehow connect. I mean, like we in our societies, we don't live on our own. If something happened to my neighbor, I know about it. I sometimes live through all the problems as my neighbor lives, even though I'm not directly affected. So going back to the gene, it sits on the X chromosome and has its own structure and has a few what we call exons, and and it gives rise to a protein with a certain structure. And I'm not going to talk about the function about PCDH19, but as it was mentioned before, it's a molecule which is quite, perhaps what we speculate, important for cells to connect and to know that they are next to each other. The cells need to know somehow who is next to them. They need to have a way how to recognize that they are in the right spot. That because during development, when the neurons and when these cells are moving, especially in our brain formation, they have to go to a particular area, and they need to know that they reach their destination. And protocaine 19, of course, we looked, it's important for guiding these cells into their right destination. Now, the spectrum of different mutations or the genetic problems which we see in protocaine 19 is really growing dramatically. And here we perhaps captured only what has been published, and I know even from my colleagues in Adelaide that there are many more mutations. Generally, we can split them in two categories. One, which completely remove the protein. So you pretty much don't have any of this molecule available for the cell. And those are the mutations which are at the bottom part of that protein structure, which is in the middle. And the other type of the mutation which are above are those where the protein, we don't know it for sure, but we can really predict with high probability, the protein is likely there, but doesn't work properly. It's interesting that when we look at clinical presentations of those individuals which don't have any protein and those which have those missense mutations, it doesn't seem to be really a major difference, which led us to postulate that even those mutations on the top, which just change small tiny bit of the molecule, are in effect the same, complete loss of function, like if you don't have the molecule at all. I mean, that's important. It's important for the reasons of modeling the disease, when we try to understand how this works in cells or when we try to make animal models. So that's very important. Collection, that's like you heard about the clinical presentations. It's hugely important. Sometimes one or two special patients can inform us hugely about where to look and what to do. So we did look, as part of our study a few years back, on the expression of the PCDH19, because just by knowing where the gene is expressed and what time during development, we can learn, or we can guess a little bit better what its function is. And you heard about the frontotemporal types of epilepsy and the frontal lobe on this top panel. You can see it here. The huge signal is there. The protein and the gene is present, it's actively engaged, it's expressed, it's there, and you can see it in other areas like hippocampus, in thalamus as well. You can see generally in cerebral cortex or even in amygdala, which is a very important part of the brain with respect to the neurological, neuropsychiatric, neurobehavioral features which you see in these patients. And then, of course, on the right-hand side, this is more through the sections of the mouse brain. You see that it's expressed in the right areas, and then within the areas in a very specific cell populations. So that's where I'm telling you about that this molecule is important for the cells to know who they have next to each other. That's where they actually end up, because they migrate, they move from one spot to another spot, and they need to know where to stop. While we don't see in these patients any major structural problems like magnetic resonance imaging, probably these cells may end up wrongly connected. We still don't really know, but it looks like there's no major problems. It's probably going to be in the connectivity, or perhaps the cells end up in slightly different layer of our cortex than they should be. So, to a little bit to our work, finally, what we have been doing and how we were going about that. It has been quite a difficult journey, I have to say. Like, it was a difficult journey to find a gene initially, but to understand how it works. It seems to be everything going completely the opposite direction, like with the inheritance. That the boys who don't have the gene are fine, and the girls who still have one properly functioning are not. So we have started to study really these problems using different tools, and that's as we do in laboratory science. We studied the cells in culture. We use the technology, I have it written as Y2H, which is the yeast-2 hybrid, where we're looking at its partners. As I said, you sometimes can tell about people based on who their friends are. So we're looking for the friends. We're looking for the other molecules involved. We also have done these high throughput technologies, which is looking what happens in these cells all together. So we're looking at all the genes, we look at the metabolites. We also had to generate tools so we can visualize these proteins, and that's why we generate the antibodies. And we also generated a mouse model who perhaps can help us to understand. Today I'm really just going to talk and touch more about the cell culture and the microarray studies, what we have done. So before start, we started to use skin cells. Why skin cells? Why not blood cells? The simple reason was that this gene we found when we looked at was present in the skin, but was not present in the blood. So that was the simple reason. And we wanted to study the effect of some of these mutations. So we started to isolate the skin. And this is not very invasive, but it's a lot more invasive like getting a blood. You need to get a piece of skin and grow it in a culture. We also started to collect skin cells because, as you know, maybe at least some of you, this year's Nobel Prize was awarded to a group in Japan and United States for the ability to generate stem cells or pluripotent stems, induce pluripotent stem cells from human skin, and then to convert those cells into different types of cells. Our problem generally in laboratory is that we want to study especially neurological disorders like epilepsy, intellectual disability, that they don't have the right cell type. And to really study the brain based on blood, it's good in certain cases, but not always. So hugely advantage is to convert the fibroblasts into, of course, the stem cells. And that's what we are doing at the moment. We don't have the cells available. And Ricardo Dolmek was going to tell you about that. He's expert in this area, and he obviously would be able to tell you more. We are making those cells, and we are making progress, but I'm not going to show you any results using those cells. It's a slow process. It's not an easy one, but eventually we end up with neurons of these patients, and that's going to make a big difference for many reasons, not just to understand what the function of the protein is, but how we can change it or rescue it in the future. So I'm going to tell you a little bit about the microarray studies, what we have done. So we have done two groups of girls. We have done it in two different time points. Originally, we started on the left-hand side with a group of 6, those EFMR girls. So the girls from the families, the large families we originally studied back in 2007, 2008. And we have taken we call them transmitting boys, or these are the boys which are from these families, have the mutation. And then we have control boys, and then we have control women as well. And then the second group was the group from Carla Marini, her collaborators, where we had the girls which have the more like the early onset, perhaps the isolated cases, I would say, if I box it together, I would say perhaps the more severe form. And they were younger as well. And we have used that second set kind of as a replication study, because we have made some findings on the original set, and then we just had to just reinforce that message by doing the second set. So what we have done, we have isolated the genetic material, the RNA, from these cells, and we used sort of the technology which can measure the expression of these molecules across the entire human transcriptome. So we measured the level of expression of about 15 to 20,000 different molecules in each one of these girls and the controls. And then we started to compare, because we wanted to know what is different. On average, we're always going to find different, but because we had so many girls with the PCDH19 mutations and controls, we could statistically test it and see whether these differences are meaningful or they are just random. So we found, as you can see down here, a different number of genes which are different according to certain criteria, and that's usually a statistical significance. overall, when we look at types of molecules which are not expressed properly or are not expressed same as in normal individuals, we found molecules which are important in cell adhesion and integrin signaling, which again makes sense, because if this is affected and the PCDH19 itself is a cell adhesion molecule, then we are probably on the right track. Interestingly, though, we found other molecules which showed big difference, and this is one example of a glutamate receptor. You heard glutamate receptors are very important molecules in the brain. They have been associated with learning disability and epilepsy, and in particular this glutamate receptor, GRIA1, is a gene which has not been directly associated with schizophrenia, but indirectly quite frequently. And as you can see, the right-hand side, the pink signal, it's the signal in the normal girls, and those two smaller bars on the left-hand side, that's the significantly reduced expression in these two different cohorts of girls with PCDH19 mutation. What was though very interesting was that, of course, we were looking for the differences between the affected girls and normal girls. But what we really found was very intriguing. And that's to some extent described on this graph. So if this is just the level of expression, so these would be the girls which are unaffected, and these are the boys which are normal. And this is the expression of a certain gene, A. So girls usually have more of it, and boys have less. Interestingly, the affected girls, as you can see, clustered more with the boys than with the girls. And that really started to ring a bell, because when you look into gene expression between boys and girls, there are differences. We call these sexually biased genes. These are usually genes which are not on the X chromosome, clearly, because women have two, and there are many of these genes which are expressed from both copies. But there are also genes which are under control of steroid hormones, which show differences between boys and girls. And the human brain is probably the biggest example of the sexually dimorphic organ, where there are huge differences between boys and girls. So in a way, here we had the expression by measuring genes in these girls with PCDH19 mutations, which looked more like the expression of the boys rather than expression of the girls. So that was very surprising, and we had to test it somehow. So we have done different statistical tests and compared the first lot, second lot, and then we combined the lot against the pool of the fibroblast controls. But we really found very significantly, statistically significant measures that this was not just by chance. So clearly there was something, or the consequence of the loss of PCDH19 function in these girls led to the dysregulation of the expression of genes to that level in these cells that they look more like boys than girls. Now that really still doesn't help very much. So we had to go back and we went to look at individual genes, that's really whether this is true, and we spent almost a year and a half to test this. So this is an example of, again, another gene. Actually, this specific gene is involved in neurosteroid hormone metabolism. So that's why we looked at it also very carefully. And again, you have one lot of the girls, second lot of the girls. These are the control girls, and these are the boys. And again, you can see the significant reduction of the expression of this gene. We looked also at the level of the protein, because sometimes on the level of the RNA is not very informative. So we need to look at the protein. And here it gets a little bit more complex, where you can see again in the girls from the first lot, slight reduction of that band. It's even missing. And here you can see it. So this is the more severe cases. These are perhaps the less severe cases. These are the familial cases, while these would be more the isolated cases. And then you have controls and other controls and the transmitting males. Interestingly, in the transmitting males, this goes slightly up. I don't have them here, but they have it slightly up. Not really statistically significant. So you can see the variation there. We haven't looked whether that variation in that specific enzyme would be able to predict to us whether that girl is more or less severely affected. Of course, that's the first question, what we would have. But we can't say it as yet. We haven't done, or, well, we're not actually actively looking into it because simply we don't have enough cases to have statistical significance in it. So really this significance in gene expression led us to really speculate that maybe there are steroid hormones involved. So then we started to look, I want to go back. So yeah, are they steroid hormones? Because that's the only perhaps plausible way or the most obvious way how to explain such gene expression differences. So here I borrowed a picture from one nice review published about in 2008, which shows the expression of different hormones, testosterone, estradiol in males and females across the development. So from birth, the 5 months, then 2 years, 5 years, 10 years, and goes there. And you can see that after birth, when the level of the hormones is very low, there is this small peak, which is called mini puberty, which stops really on average sort of about 6 to 9 months of age, but it varies significantly, especially in girls, can go up to 2 years. And then you have the time when the expression levels are very low, and then during puberty, of course, the hormones go up. So what we started to do, we started to plot the age of onset against this from the girls. And when we looked quickly from our study, which is the study of Ingrid Scheffer from 2008, the onset of the more like EFMR girls were about 14 months. And the offset, so where the seizures stop, because these girls and women are old enough, so they already know where sort of the epileptic activity stopped. So that's why we know the seizure offset. And the offset was median, so the average offset was about 12 years. So it's when the puberty comes. Then when we looked at the study of the French group, Despien et al., then we saw the onset a little bit earlier, but sort of not very different to the girls. And then, of course, when we looked at it, we saw the major seizure activity happening really within the time where the hormones were low. So my student collected data from the literature on all so far published cases with PCDH19 mutations and put it against that chart. You can see it's slightly modified. So those are the circles, red dots. And the blue triangles are all those girls where we know that the seizures stopped. And when you really look at it and put all these data together, we get the median, or the mean, I should say. It's mean onset about 10 months, and I think that fits well with what Carla and other speakers presented today. And then we get about mean offset, so when the seizures stop, about 12 years of age. But what is important also to see that the distribution is quite wide. You have some girls which really don't start until quite late, but some girls which stop already by then. But at least to me, this makes perfect sense if steroid hormones were involved, because the variation in onset and offset of puberty and the steroid hormone levels is so dramatic that that can be the major modifier of this, if it's the case. So we really kept looking into more and more and tried to find more sort of evidence that we are on the right track. So we took also the example of the catamenial epilepsy, which was mentioned today as well. It's a very good example of a type of epilepsy which is due to the variation of steroid hormones. And as you can see, this is from a nice study of Reddy et al. Published about 3 years ago. The highest frequency of epilepsy during the perimenstrual cycle, so where the hormone levels are very low. And also there is another type, the ovulatory type, which is sort of at a time where the allopregnanolone levels are very low. So that's also to some extent encouraged us that most likely these type of molecules might be involved. Now, when you look in the literature, and this is from the same work of Reddy et al. Published, there are many neurosteroids which are really has been tested or showed that they have anti-convulsant, so anti-epileptic effect. But importantly, there are some which have pro-convulsive effect as well. And as Carla mentioned, allopregnanolone, even progesterone, dihydroprogesterone, and all of these are listed with anti-convulsant effects. So that, to some extent, would make sense as well. When you look molecularly what these molecules are doing and how they affect their function, sometimes it's via regulating of gene expression, and that's one good example through progesterone and progesterone receptor, and then regulates neuronal gene expression. Or it's via direct effect on the important molecule which are really crucial for epileptogenesis, and they are the GABA receptors. So the function can be either regulation of gene expression or through directly looking at the channels which are important in the electrical activity of the brain. Now, what is interesting that people who published knockout mice, so the mice without progesterone receptors, the PCDH19 molecule is downregulated. So actually it's very low. And these mice are also less resistant, or so they are more resistant to certain type of epilepsy. So again, that suggests that the progesterone and this pathway, it's quite important for that type of epilepsy. When you look at really the very small level of connection between the neurons, and that's a level of the synapse, and this is the presynaptic, this is a postsynaptic, and then there is glial cells or astrocyte. There's a lot of these molecules expressed here. We don't know, and Carlo probably will tell you more about where PCDH19 is expressed. But certainly the neurosteroids, which are generally generated either through mitochondria from cholesterol, are very important in controlling some of this action. And not just from the neurons, but more importantly also from the non-neuronal cells. And so this is all happening. In terms of expression, these molecules are expressed in the right place. So again, just by the fact that they are present there, we can implicate them that they are likely also implicated, but it still requires a lot more work for us to be really sure that these molecules are the ones we would like to target in this group of disorders. So if I want to summarize for you what I was trying to tell you is, and what other people are also trying to tell you, is now PCDH19 is now recognized to be actually among the most frequently mutated genes in epilepsy. How to call it? I call it here as a PCDH19 female-limited epilepsy. Of course, it's not only female-limited because there is at least one male, and probably we're going to see more, but it's primarily females. Name is important. That's how people remember lots of things. So whether it's a PCDH19 FLE or a PCDH19 spectrum, or I'll leave it to the others to sort it out. Now, the clinical expressivity is really highly variable. But what is also important, we have these non-expressing girls, which are almost unaffected. And I think they are very important for us to study as well, because they can tell us about what actually protects them. Usually, you don't get to see these girls, of course, because they don't see the doctors. So we normally ascertain them through the families. There are the grandmothers or mothers or somewhere, older ones, because you find the daughter or granddaughter, and then you go back and you track the mutation in the family. You suddenly find that there are one or two girls. And these are really important, because by studying these girls, we can really understand what protects them. What is it? Is it the hormones, or is it something else which makes them not have problems like the other girls in their family? So in our studies and what I showed you, and we have a little bit more evidence from the other aspects of the spectrum of experiments, what we do for support of the involvement of neurosteroids, but it's still, I still call it a hypothesis rather than something we would like to invest a lot of study. We still need to do more before we believe that that's correct. And I think the steroid hormones also can explain the clinical expressivity. At least I believe they are a very good candidate to be involved. And there have been lots of studies, Carla mentioned, of steroid hormones in epilepsy. The only problem is when those studies are conducted, that they are conducted on patients without known genetic mutations. So that's why the outcomes are not always clear. Like you take 10 patients with epilepsy and you treat them with particular neurosteroids, and 2 patients will get better and 8 of them don't. But the problem is that we don't know what is really the cause of the epilepsy in those 10 patients. So as soon as we can really take only those specific patients which are defined by their cause and then conduct studies using these molecules, then we'll be able to say, again with more certainty, but not with definitive answer, that these molecules will help. I believe that they likely will help, but there's still a long way to go. So there are many questions which remain, and you're probably going to have many more. So clearly, where do we go from here? Do we focus on those neurosteroids, or do we focus on glutamate receptors? I showed you that one of the glutamate receptors have been significantly deregulated. There are other very interesting molecules. So clearly, the steroids are not going to be all. There's going to be probably only part of the picture. There are going to be other aspects of this phenotype. What if, let's say, if we're going to be able to fix something with the steroid, the question is what that's going to be. Is that just going to be the seizure activity, or it's going to be more? We still don't know really what is the normal function of this molecule, whether it's really just to know that the cells are next to each other and where they're meant to go, or there is something more to it. And especially during which particular stage of the development these molecules are crucial. And this is what I mentioned, why some of the girls with these mutations are less severely or not affected at all. Now, important factor is, and you heard it today, and that's really for the discussion as well, what is the role of the environment and perhaps stress? We talk about infection, vaccination, the fever. I don't know, sometimes stress apparently can protect from epilepsy. Sometimes it can trigger epilepsy. So it's really complex, but the question is what really starts it? And how important it is when it starts, how it's going to go. So these are really very important questions, and we don't have answers for them. And probably you have many more. So I think I will stop here and let the other speakers to continue, and then we can discuss. Thank you.